Greetings and warm regards. Myself, Dr. Priyanka Bhanushali, third year postgraduate in Department of Oral Pathology and Microbiology at Ahmedabad Dental College. My topic for today's presentation is saliva as a diagnostic window to the body. The presentation will be covered under the following headings, starting with introduction, saliva sampling, salivary diagnostic tools, diagnostic applications of saliva for oral and systemic diseases, forensic evidence through saliva, and point of care saliva testing. Saliva has been studied extensively as a potential diagnostic tool over the last decades due to its ease and non-invasive accessibility, along with its abundance of biomarkers like genetic material and proteins. Salivary diagnostic holds a great promise as an effective modality for early diagnosis, prognostication, and monitoring the post-therapy status. In this review paper, we have emphasized role of salivary biomarkers as diagnostic tools and hence saliva as a diagnostic window to the body. Advantages of using saliva are innumerable. It is a non-invasive, easy-to-use, inexpensive diagnostic tool, safer than serum sampling. It gives real-time diagnostic values. There is no need for trained medical staff. Multiple samples can be obtained easily. The collection and screening can be done at home. Has minimal risks of cross-contamination, is more economical, and requires less management. Saliva sampling. Saliva collection can be done by two methods, collection of whole saliva and saliva sampling from different salivary glands. These are various diagrams showing the various methods like draining method, spitting method, various swab-based devices for sampling of the saliva, graduated tubes or ductal and suction tubes, various micropipettes which can be used for saliva sampling. Lastly, the Lashley cup for parotid gland saliva sampling is also shown. After collection, the processing and storage of saliva is of utmost importance. Chemical stability of the biomarker and as well as the diagnostic purpose determine the way sample should be processed. Collection is basically into ice-cooled vials. Also, the collection can be done in vials containing cocktail of protease inhibitors including EDTA or others. Bacteria and cellular debris have to be removed directly after collection, which can be done by centrifugation or filtration. When it is not possible to keep saliva as frozen, often sodium azide is added to retard the bacterial growth. After collection, samples must be snap frozen, preferentially in liquid nitrogen, especially when samples have to be stored for a prolonged time period. Storage at minus 80 degrees Celsius is preferred over minus 20 degrees Celsius. To maintain integrity of the proteins, deep frozen sample must be thawed as quickly as possible, like by transferring the sample vial to hot water for a short period of time. To avoid repetitive freezing and thawing of saliva samples, they can be stored in 100 microliter aliquots. Coming to salivary diagnostic tools. Salivary diagnostic tools comprises of cylometry, cylochemistry, and various subparts of the salivary omic technologies, that is genomics, transcriptomics, proteomics, metabolomics, and microbiomics. Cylometry allows the volume measurement of saliva in standard conditions for whole and gland-specific saliva in stimulated and unstimulated conditions. Its use is in conditions like hyposalivation, which is difficult to diagnose otherwise. Hyposalivation may be due to diabetes mellitus, hypothyroidism, dehydration, or any other causes like radiotherapy of head and neck region, etc. In these cases, monitoring the saliva volume by cylometry would be of utmost importance. Cylochemistry examines the electrolytes and proteins in saliva. Its applications are in determining and monitoring level of various hormones like cortisol, progesterone, testosterone, for monitoring drug levels like diazepam, caffeine, lithium, etc. Saliva may also be used to monitor patient compliance with psychiatric medications. Saliva omics. It is a broad collection of technologies used to explore different types of molecules contained in saliva. There are 5 major diagnostic alphabets available in saliva, namely proteins, genetic material, DNA, messenger RNAs and microRNAs, metabolic compounds, and microbes. The saliva omics includes genomics, that is study of genes and their methylation, transcriptomics, the study of mRNA within cells or organisms, metabolomics, that is the study of global metabolic profiles in a system, proteomics, that is study of proteins, and microbiomics, the study of microbiome. Salivary genomics and epigenomics. The salivary genome consists of both human and microbial DNA. A proportion of roughly 70% is of human origin and the other 30% is originated from oral microbiota. Saliva samples also yield sufficient DNA for sequencing arrays and PCR assays. The quality of salivary DNA is comparable to that in blood. Salivary transcriptomics. Whole saliva and saliva supernatant harbor both microRNAs and total RNA. Investigators probing the salivary transcriptome have identified over 1,000 microRNAs and more than 3,000 species of mRNAs in human saliva. Composed of coding and non-coding RNAs, the transcriptome makes a significant source of potentially relevant diagnostic information with highly specific discriminatory indicators. Salivary proteomics. The salivary proteome comprises entire protein content of the oral cavity. It contains more than 2,000 proteins and peptides. Salivary proteins are involved in a number of metabolic pathways, and researchers have reported unique proteomic profiles indicative of oral cancer, endocrinological and connective tissue disorders, caries, periodontitis, and even breast carcinoma in saliva. The analysis of salivary proteomes may reveal morbidity signature in early stage and monitor disease progression. Salivary metabolomics. The metabolome validates parallel assessment of a group of endogenous and exogenous metabolites like lipids, amino acids, etc., which is valuable tool to discover biomarkers for monitoring physiological status and making proper treatment decisions. Salivary microbiomics. Due to constant microbial replacement, salivary microbiota reflects many changes occurring in any part of the mouth as a fingerprint of whole microbiota. The recent advances in NGS has allowed identification of about 19,000 phylotypes in oral cavity. Evidence shows that bacteria and microorganisms can lead to oral diseases, which can be monitored by salivary microbiome. Saliva as a tool for liquid biopsy. Liquid biopsy refers to analytes from various biological fluids like blood or other accessible fluids. It is a non-invasive diagnostic tool. One of the key advantages of studying liquid biopsy is that they provide personalized snapshot of primary and metastatic tumors at successive time points. It also provides molecular profiles for each patient and offers great advantage of being less stressful. Currently, most common analytes used are circulating tumor cells, cell-free tumor DNA, metabolites, and exosomes. Coming to diagnostic applications of saliva. Starting with oral diseases, biomarkers for dental caries can be of two types: caries risk assessment by analyzing levels of cariogenic bacteria in saliva, or caries risk assessment by analyzing host-related factors in the saliva. Biomarkers for periodontitis. 6 clusters of markers released during immune response may be eligible as biomarkers for periodontitis. These include host-derived enzymes, tissue breakdown products, host response modifiers, cytokines, oxidative stress markers, and specific and non-specific proteins. These all clusters of markers are released during the interaction of host with the bacteria and hence can be useful predictors for periodontitis. Oral potentially malignant disorders should be under constant surveillance because they have the highest chance of malignant transformation. Various salivary biomarkers can be used to detect its progression to cancer. Various genomic studies for DNA aneuploidy, mutations in p53 gene, or presence of HPV and EBV genomes can be used because they have an increased association with oral cancer and are also linked to advancement of pre-cancer to cancer. Various mRNAs, microRNAs, and proteins can also be used as salivary biomarkers for the same. Oral squamous cell carcinoma. For oral squamous cell carcinoma, saliva could be the most important diagnostic tool because it also contains the exfoliated cells in oral cavity, which would allow saliva to be the first choice of screening and identification for potential biomarkers for oral cancer. These biomarkers include various changes in cellular DNA, altered mRNA transcripts, and altered protein markers. Role of salivary biomarkers in other malignancies. Various distal malignancies can also be detected through salivary biomarkers. These include pancreatic cancer, breast cancer, gastric cancer, ovarian cancer, and lung cancer. Coming to various systemic diseases which can be monitored and diagnosed through saliva. Various cardiovascular disorders can be monitored or detected using saliva. Literature has been published on importance of salivary biomarkers, and these biomolecules have a positive correlation between serum and salivary levels. These include myoglobin, cardiac troponin, CKMB, myeloperoxidase, to name a few. For diabetes mellitus, monitoring of salivary glucose, adiponectin, and alpha-2 macroglobulin levels can act as biomarkers for monitoring the disease. These components have a positive correlation between serum and salivary levels and hence can act as putative biomarkers. Various bacterial infections, parasitic infestations, and fungal infections can also be detected using saliva. To name the viral infections, hepatitis, measles, mumps, rubella, dengue, HPV, COVID-19 can all be detected using saliva. They can either be detected by presence of IgM antibodies against the virus or by performing PCR on the genetic material available from saliva. Saliva has also been proposed for monitoring of systemic levels of the drugs. It's useful for monitoring anti-epileptic and anti-cancer drugs. Various other drugs can also be monitored through saliva, and various abuse or recreational drugs like amphetamines, cocaine, nicotine can also be monitored. Saliva can also be analyzed as a part of evaluation of endocrine function. Substrates monitored include salivary androgens, salivary progestins, estrogens, salivary glucocorticoids, etc. Applications include psychobiology, sports medicine, pharmacological studies, pediatric studies, etc. Salivary cortisol is used to investigate adrenal function, and recently there has been a considerable interest in use of bedtime salivary cortisol levels as a screening test for Cushing's disease. Saliva as a forensic tool. Saliva can be used for analysis of drugs of abuse in saliva, cigarette consumption detection, organic substance and heavy metal intoxication detection, personal identification, sex determination, bite mark analysis, and blood group antigen detection. Point of care testing. It is defined as medical testing conducted outside a laboratory at or near the site of patient care, including patient's bedside or doctor's office or patient's home. The salivary diagnostic toolboxes include proteomes, metabolomes, genomes, microbiomes, and immunologic categories. All these toolboxes are used for different diseases and employ different technologies for the same. These are various point-of-care devices for detection of diseases through salivary biomarkers, along with the technology used. Many emerging novel point-of-care technologies include smartphones, microfluidic platform, electrochemical biosensors. In conclusion, salivary diagnostics is a dynamic field that has been incorporated as a part of disease diagnosis, clinical monitoring, and to make important clinical decisions. The real promise of salivary analysis use is the ability of patient or clinician to directly and continuously assess the disease status, progression, and therapeutic efficiency. Hence, truly, saliva can be considered as a diagnostic window to the body, which can reflect virtually the entire spectrum of normal and disease state. These are some of my references. Thank you very much.